So now that we have learned the basics of Punnett squares and how to predict the chances that offspring will have certain genotypes or phenotypes, let's talk about some other things we can do with this information. What are some other things we can do with making Punnett squares and with genetics and all that? One of the reasons in the real world we would use something like this is in the field of genetic counseling, which kind of ties into some of the stuff we've talked about already. Remember that in humans we have these 46 chromosomes, and that each one of these sets of chromosomes has the alleles for certain traits. Sometimes there could be genes on these alleles that encode for genetic disorders. Genetic disorder is any abnormal allele, and it's basically going to cause some sort of defect in the normal gene. Some of these alleles could be on autosomes. So if there's a defect in a gene that's on an autosome, remember autosomes are set 1 through 22. So if there's a defect on any one of these chromosomes, we call this an autosomal disorder. So once again, an autosomal disorder is there's gene defects on chromosomes anywhere in set 1 through 22. Now there's other disorders that are carried on the sex chromosomes, and these would be called sex-linked disorders. So again, sex-linked disorders if there's a genetic defect on one of the sex chromosomes. And it just so happens that most of the time the defect is on the X and not the Y. Because actually Y chromosomes don't really do much at all. It's the X that has most of the important traits. In today's lecture, we're going to focus just on this first set, the autosomal disorders. And in a couple weeks, we'll talk about the sex-linked disorders later. So today we're going to focus here on autosomal disorders. Now here's something to note. Most autosomal disorders are recessive. So let me kind of show you what that would mean. Let's say, for example, we had this gene F, and F stands for the normal version of the gene that's functional and there's no problem with it. And then we had this little f, and the little f would code for the defective form of the gene that causes a disorder. So very clearly, if somebody was homozygous dominant, they would have this normal functioning gene, no problem, because they have two normal copies of it. If somebody was heterozygous, so they had one of each, even though they have one copy of a defective gene, the normal gene is dominant. So in this case, they would also be normal. However, they would be a carrier of this genetic disorder because they're carrying one copy of the defective gene, which means they could pass that defective gene on to their children. Then lastly, of course, if we had a homozygous recessive individuals that had two copies of the defective gene, then this individual would have the disorder, whatever we're talking about. So again, when I say most autosomal disorders are recessive, what that means is in order to actually have the disorder, usually the person has to have two copies of the defective gene in order to actually have the disorder. If they have one, they could just be a carrier. Now, a lot of times in genetic counseling and just in genetics in general, it's very easy for us to make something that looks like this. This diagram is called a pedigree. And the easiest way to put this is a pedigree looks just like a family tree, but it's a family tree for genetics. So basically, it's your genetic family tree. And it's a way for us to visualize whether or not certain defective genes could spread to offspring and the probability. So whenever you're going to make a pedigree, whenever you see squares in the pedigree, these represent XY males. Whenever you see circles, these represent XX females. So for example, this circle is clearly an XX. This square is an XY because it's a square. Now the other thing that you'll see in these, some of the squares or circles may be shaded in. So if the square or circle is fully shaded, this means the person has the genetic disorder that we're talking about. If there's no shading at all, it means the person has two normal alleles for whatever genetic disorder we're talking about. If you see a half shaded, and we don't always do this, but I think for this class, I'll just make it easy and I'll say, if you see a half shaded, this means the person is a carrier which would indicate they are heterozygous. They have one copy of the normal allele, one copy of the defective allele. Now, when you see lines like this that are connecting one circle to a square, this indicates this is the individuals that are going to mate with each other. So, for example, this could be somebody's mother and father, or a sperm and egg if it's a donor situation. So this is going to be the egg and sperm that mate. So horizontal lines indicate a mating between an egg and sperm, and vertical lines indicate the offspring for that mating. So, for example, if this egg and sperm were to mate, these individuals down here are their possible children, their offspring. And then since this circle is not directly linked to these, you could tell this is an egg that's going to mate with this offspring sperm, right? And then here's another vertical line indicating this individual's had this child. So that's basically what a pedigree would look like and all the different features of the pedigree. Let me give you an example of an autosomal recessive disorder. One example is a disease called cystic fibrosis. Cystic fibrosis is an inherited disease, and this individual needs to have 2 copies of the defective gene. What it leads to is there's a problem in one of the membrane transporters in the cell membrane. So the cell releases an abnormal amount of mucus, a lot more than a normal cell would. And this mucus can build up in the respiratory airways and in the lungs. Normally, having a little bit of mucus is good. It can trap bacteria and help the person cough and get rid of that bacteria so the bacteria doesn't get to your lungs. But too much mucus is bad because now bacteria and viruses can get trapped more easily in the respiratory airways and lead to infection, severe infections in the lungs that normally wouldn't affect a healthy individual. So historically, individuals with cystic fibrosis have had a very young average age of death, which, as you can see, in the 70s, it was basically like at birth because there was no treatments. As we've developed treatments over time, now these treatments are not cures. There's no really cure for this disease because it's genetic. It's in the cells, it's in the nucleus. But the treatments that are used are ones that can help basically thin out this mucus and make it so it's less sticky, make it so hopefully the cell doesn't secrete as much mucus. And basically the individual has to try to be very careful staying away from anybody who has an infection so that they don't actually catch the infection. So basically help the patient's immune system as much as possible. So because of that, now the average age of death is over 30. Actually, I had my best friend in childhood had cystic fibrosis, and she's 33 now. She's still alive. She had to get a double lung transplant because her lungs were just so damaged from so many years of infection. So this is one of those unfortunate autosomal recessive disorders. Another one is called Huntington's disease. Huntington's disease actually affects the neurons in the brain. So again, this is an inherited genetic disorder. But this one is actually autosomal dominant, which means the individual only needs one copy of the defective gene to get Huntington's. And basically what Huntington's does is it affects the neurons and it causes a severe loss in neurons even at a very young age. So almost like a very early onset dementia symptoms will develop. So let's walk through a process that a genetic counselor might actually do if they wanted to test the chances of a genetic disorder developing in an egg and sperm, right? And let's talk about cystic fibrosis. So let's say that capital F is of course normal, and a lowercase f would indicate the cystic fibrosis disease. So let's say we had this egg, it's a circle, so we know that it's the egg. And let's say that this individual knew they were a carrier, so they had one copy of the big F normal allele and one copy of the cystic fibrosis allele. And let's say this individual was going to mate with this sperm. And in this example, let's say that this individual also knew they were a carrier of cystic fibrosis. So they are also heterozygous and would have one big F and one little f. And this couple wants to know their chances that their offspring will have cystic fibrosis. So what you're going to do as always is make your Punnett square. You'll put your egg here on the top. So we know there's one big F, one little f. We'll put our sperm alleles here on the side. So again, there's one big F and one little f. Okay, now we're going to cross these alleles. So now after we've crossed the alleles, we're going to look first at the genotypes. So if you were a genetic counselor for this couple, you could tell them that they have a 25% chance of having a baby who has two big Fs, which means homozygous dominant. And then this couple might say, what does that mean? And you would tell them if there's two big Fs, homozygous dominant, the phenotype would be no cystic fibrosis. Next, this square and this square are heterozygous. So there's 2 out of 4 chance, that's 50% chance of having one big F and one little f, which is heterozygous genotype. And then of course, phenotypically, that would mean there's a 50% chance this couple will have a baby who's a carrier of cystic fibrosis. And then lastly, there's a 25% chance that this child could have two little f's, which would mean homozygous recessive, which phenotypically means does have cystic fibrosis. So overall, what you could tell this couple from their genetics, based on the fact that they're both carriers, that there's a 25% chance that their offspring will have this genetic disorder. Now, the last one to talk about is sickle cell anemia. Sickle cell anemia is also an autosomal recessive disorder. We've talked about it before, but let's review what sickle cell is again.
In order to understand this process, we have to know about hemoglobin. Now, red blood cells normally have this round shape, but they have kind of this biconcave center. It's like a little dip in the center. It's kind of like a donut that they didn't take the hole out in the middle completely. Inside the blood cells, there's this molecule called hemoglobin, and that molecule is so important because it contains iron. And that iron is what binds to oxygen and carries oxygen throughout your body so that all your muscles can get the oxygen they need to make ATP. Now, there is a genetic disorder. It's called sickle cell anemia. Someone with sickle cell anemia, their blood cell takes on a sickle, or it's kind of like a crescent moon shape. When blood cells are round, they can flow through capillaries very nicely and smoothly. But when they have this sickle cell shape, they potentially could get lodged in the capillaries and block blood flow. And also, sickle cells have these abnormal hemoglobin molecules in them. The genotype, the allele of a normal red blood cell, is HB, that stands for hemoglobin beta A, but the allele for sickle cell is HBS, S for sickle. The only thing that makes a sickle cell different from a normal red blood cell is one single amino acid mutation. One single glutamic acid has been mutated to a valine. If a person inherits two normal red blood cell alleles, two HBAs from their mother and their father, they're going to have a normal red blood cell. If the individual inherited one copy of each, so they inherited one HBA from either their mother or father and one HBS, they're going to be what we call a carrier. They're not going to have sickle cell anemia. They're going to just be a carrier of the trait, which means they could possibly pass that gene to their children. In order for someone to have sickle cell anemia, they need to have both. Both copies need to have the sickle cell trait. So that means they needed to inherit a sickle cell gene from their mother and from their father. In order to predict if someone potentially could have sickle cell disease, just like always, we'll do a Punnett square. So if you knew egg was a carrier of sickle cell, so they had one normal and one sickle cell gene, and we knew the sperm was also a carrier, we would make a simple Punnett square, and what we would see is 25% chance this couple could have a child with a normal red blood cell. There's a 50% chance the child could be a carrier, and there's a 25% chance that their child could have sickle cell disease.